Have you ever wondered why, after decades of research, we still don't have a magic bullet, a magic drug for sepsis? Why do some patients recover quickly while others spiral into a multi organ failure and eventually die? Now, this is despite the best of care that we give in our ICUs. Now, we frequently look for answers with latest antibiotics, trying to look for microbial antibiotic resistance, how to overcome them. What if the answer lies not in these microbes, but in our own immune system and its unpredictable behavior? In this particular video, we will do a deep dive into sepsis, not just the emergency that we deal with, but as a complex story of immune chaos, medical challenge, and the future of personalized critical care. Now, whether you are an MBBS student or a ICU specialist, there is definitely something for everyone here. So let's start with the basic. Sepsis is not simply severe infection. It's life-threatening syndrome where the body's own response to the infection spirals out of control, causing acute organ dysfunction. Every year, nearly 50 million people are affected, with almost 11 million losing their lives to sepsis globally. Now, here's the catch. The real danger isn't always the microbe, because these microbes are everywhere. A much larger number of people are getting affected with these microbes, but not everyone is developing sepsis. So it is our immune system's response which is at flaw. In sepsis, there is a simultaneous explosion of inflammation, that is cytokines, hyperactivated immune cells, and paradoxically, there is also a collapse of the protective immunity, making patients vulnerable to secondary infections as well. So some recover fast, while some will linger for weeks. Some will succumb early, while some will die late due to the long-term complications. Now, why is it so unpredictable? Because every patient's immune response is different. Comorbidities are different. The organism is different. Age is different. The genetic profile of the patient is different. Even the timing of the immune response. Now, all this combined to create an unpredictable and a dynamic disorder. So let's break it down further. In the earlier stages of sepsis, the innate and the adaptive immune system, they both go haywire. Macrophages, neutrophils, the natural killer cells, they release massive amounts of cytokines. Some trying to fight the invaders, others trying to put the brake on the inflammation. This leads to tissue injury and organ dysfunction. At the same time, immune cell apoptosis, which is programmed cell death, kicks in, leading to what is known as immunosuppression. This is why many patients, after surviving the initial storm, die weeks later from secondary or opportunistic infections. This is extremely frustrating. When you are in the ICU, you give all your effort, resuscitate the patient, make them survive, and eventually they develop a secondary infection and die. So to make things more complicated, the immune status of each patient shifts over time. The same cell can be pro-inflammatory at one stage and anti-inflammatory at another. In fact, the very therapies that could help one patient may harm another if given at the wrong time or immune phase. So where do we stand with the treatments? Now we know that the things are so unpredictable. How do we deal with this? Now, despite numerous clinical trials, now most of these trials are trying to go with the approach of one size fits all. This is the approach we are going with. They have all failed. Drugs targeting IL-1, tissue necrotic factor alpha, TLR4, even steroids, they all have had mixed or in some cases even disappointing results. Why? Because they don't account for the dynamic, patient-specific nature of the immune dysfunction in sepsis. So, is it all bleak? Do we don't have any hope? Or do we have anything with us? Let's see what we do have. Personalized immunotherapy. The key may lie in the immune profiling, that is classifying our patients as hyperinflammatory, immunoparalyzed, or somewhere in between using biomarkers like HLA-DR expression, ferritin, various gene signatures. Trials are now targeting treatment like anakinra or interferon gamma only to those subgroups that are most likely to show benefit. So identifying those phenotypes is of paramount importance. Next, immune checkpoint modulators. Drugs that have revolutionized cancer treatment. These are drugs like anti-PD-1, anti-CTLA-4, anti-TIGIT antibodies. They're all being investigated in sepsis. Early preliminary evidence has suggested that they may help reverse the immune paralysis and improve outcomes. But careful patient selection is crucial over here to avoid tipping the balance towards autoimmunity. Next, immunostimulatory agents. Therapies like GCSF, GM-CSF, interleukin 7, thymosin alpha 1, they can boost specific immune functions, especially in patients stuck in the immune suppressed phase. However, it is important to identify the patient is in immune suppressed phase. We have to time it, dose it, and select our patients, which is extremely challenging. Then we have new frontiers like mesenchymal stem cells, macrolides, and even some herbal medications are being studied for their ability to reset the immune homeostasis. These therapies aim not just to kill the microbe, but to recalibrate the immune response. So, what does the future actually hold for us? Instead of a single miracle drug, the future of sepsis is heading towards precision medicine. Rapid immune profiling at the bedside is important because just immune profiling is not important. You have to do it rapidly. Then give your targeted therapies and adapt it to the patient's current immune status. In this, the rapid advance of the artificial intelligence and machine learning is very important because they have helped in identifying patient phenotypes and predict trajectories for us. But how can we translate them, that is the mathematical equations, into the real world of ICU practice? This is definitely a work which is currently in progress and we all have to work towards it. The real breakthrough will come when we can move beyond just controlling the infection and supporting the organ to actively monitor and correct this immune dysfunction in real time. If you are still with me, here's the big message. The war with sepsis will be won not just in the lab or with new antibiotics, but by understanding and managing our own immune system strengths and its dangerous flaws. As researchers, as clinicians, it's time to think not just about the bug, but also about the host. So if you found this topic insightful, let me know what other aspects of sepsis or critical care you would like me to cover. Thank you for your patience.